Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is April 30th, 2026. We're talking with Dr. Courtney Rouse. Courtney is a senior research engineer at the Southwest Research Institute. We call SWRI.
Yeah, or SWRI.
So Courtney is an engineer. She's worked on a variety of data analysis problems, including some that are important in neuroscientists, like diagnosis of brain injury, self-tracking, and microscope images, as well as other ones. And she's an expert in making custom machine learning solutions for solving complicated multidimensional data analysis problems that don't have ready-made, off-the-shelf solutions that everybody is using. So, hi, Courtney, and thank you for joining us.
Thank you for having me.
Also here is Erick Olivares, computational neuroscientist and data analyst here at UTSA. Should I say something different about you? Here's what I know about you.
Yeah. Hi, Shirley.
And I'm Charlie Wilson. So, Courtney, more and more we experimental neuroscientists are collecting data that call out for analysis by some machine learning solution. And in some cases, there are commercially available packages available, and neuroscientists can just go out and buy these, or maybe they are open source and we can just download them, and then we have to figure out how to use them. And then whatever they can do, that's what we can do.
Yeah, downloading just even open source and using it's sometimes not as trivial as it sounds.
Yes. So we're lucky, I think, when we can get something and get it to work. But if we discover it doesn't quite do everything we want it to do, most of the time, we're just kind of stuck, and we have to do whatever that piece of software can do. But in addition, there are lots of solutions for which there just isn't any standard software. I mean, there's a lot of data analysis problems that don't have one of those. Mm-hmm. And the more, innovative you are, the more unusual and unique your data set is, the less likely you are to find an off-the-shelf solution. So the people that are trying to do new things are at a disadvantage, analysis-wise, for this. So we need to be able to roll our own. At least some of us need to be able to roll our own solutions to these things. So could you help just sort of lay out the landscape of choices that have to be made, opportunities that are out there, why one kind of data fits better with one kind of machine learning algorithm? I don't know.
Yeah.
As well as you can.
Yeah, I mean, sometimes I work with scientists that already have software that they're using, and maybe it performs some analysis, but not all of what they want to do. And so sometimes we have to figure out how to interface with that. And some companies make that easy, where there's plugins that you can create your own script to do whatever you want. And other companies, I'm guessing, see that as a disadvantage to them to allow you to add whatever extra analysis to things. But even if there is that available, it still takes a lot of trial and error and figuring out what other people have done and how they've used that to connect with the current software. And then, I mean, other times the doctors or scientists we're working with just have data and they haven't tried to analyze it much. And so then it's really up to us on how to get started. And so a lot of the projects I've done that are related to neuroscience are image-based. And so we're looking at feeding 2D images that are sometimes representing something two-dimensional and sometimes representing something three-dimensional. And we have to figure out what types of computer vision algorithms are going to meet the needs of the research. And if it's going beyond just the research and we're really looking to make this something that people can use in a time-efficient manner, then we also have to look into practical considerations of, like, can this run on the computer that the scientist is using?
In a reasonable amount of time.
Yeah.
So I guess most of the time in these machine vision problems, you have a thing, you know what it is that you're trying to identify in the image. And then there are other things that are not that. And you don't care about them.
Yeah.
Or at least you don't care about them as much. Or maybe you have two different kinds of things you want to identify and forget about everything else. So I'm used to thinking about kind of a template. Like I'm looking, if I'm going to track a mouse around in a cage. I know this is a solution that's already been solved by other people, but if I wanted to track a mouse around in a cage, I need to know what a mouse looks like well enough so I could identify it in every frame. So how do you isolate the features of a thing like that you want to identify? Because everything looks different from everything else. So specifically, if I wanted to find a mouse, or if I wanted to find a cancer cell in a microscope slide.
So in both cases, you need lots of training data, maybe from different angles if it's the mouse, like a front view, side view, back view. And then in the cell's case, you'd want lots of different images of what this specific type of cell looks like. And that's the data that you train the algorithm on. And a lot of times the amount of data is a limitation for us. And so we have to come up with some more creative solutions that fit the scientists or our collaborators' goals, but are not going to lead to problems down the line when the algorithm's being used, such as like overtraining the algorithm. So if you trained it on only one mouse, it would potentially learn to only identify that one mouse. And if a second mouse looked different, it wouldn't be able to identify that. And so you want to have a lot of different varying examples of all of the classes that you're trying to identify. And if that's not available, we'll do things like flip the axis of an image or rotate it so that you're working with, you don't have to find new data, but you have a slightly different data set. You know, if you flipped an image of a cell, it's still a good example of a cell most of the time. And then there's a whole nother field of research that we're starting to look into. Haven't done too much in the medical space, but it's kind of a broad application, and that's synthetic data. And so if you can create your own data using one neural network and then train your main neural network on the synthetic data.
Use generative AI to make your training set. So that's really cool. The cool thing about this, as you're describing it, is I don't have to parametrically describe a mouse. A mouse has 2 ears and 1 tail and 4 legs and all that stuff, and I don't have to do that because I don't have to explain a mouse. I'm just going to show some algorithm a mouse over and over again, and it's going to discover what constitutes a mouse.
Yeah, our brain doesn't learn like that. I mean, it may subconsciously see like, oh, the mouse has 2 eyes and whiskers and it's usually white, but
yeah, well, we've had numerous podcasts where we talk about how the brain does it. I don't think it's obvious how the brain does those things. But machine learning algorithms, at least, so somehow we turn our image into something that can go into a machine learning algorithm. So I can't just bring a machine learning algorithm up on my computer and say, look at this image, right? I've got to somehow turn that into input to something.
Yeah, an image is really just a bunch of numbers representing the pixel color.
So you need the annotated data for to telling the network this is what it is, and then it can recognize the thing.
Yeah, which parts of the entire image are that object? If we're doing like image segmentation and classification, say you want to segment out one part of the image, then you would choose the pixels and label those pixels as one object. Yeah.
And then those pixels are a matrix of numbers, and then those numbers go where? What do we do with those numbers?
That's the input data, and the computer vision model is going to look through the pixels of the input data and the surrounding pixels. So a good example of how it works would be like the border of an object. If you have a dark background and you're identifying something light, then it's going to detect that this must be the edge of the object because the pixel next door is dark and this one is light.
So that's good for finding edges. How about for finding a mouse?
So for a mouse, it's still detecting that maybe this part is a nose and that's eyes, and all together. I mean, it's not really learning that a nose and eyes, but that pattern is generally a mouse with the white background, and then maybe whatever else.
So I think there's more than one. I mean, what I'm trying for is a little bit more technical solution. Like there's more than one kind of algorithm. Some of them are statistical. Some of them use a neural network, artificial neural network. In the statistical one, I can easily picture I've got a matrix that can be the input to some statistical operation that actually operates on matrices and gives a number as a solution. And now I would have to hunt for the right way to collect all of these data and get that solution from every one of these pictures. So how does that work? Like they have names, these algorithms. I would need to pick one.
Yeah.
How would I pick one?
Reading a lot of journal papers and seeing what people have done in the past. So like the algorithm that we use to identify the unstained neuron somas was called UNet, and we knew that was originally designed for cell-based computer vision. And so it was good at identifying objects in really dense populations and very small features.
So I don't really have to know very much. I could Google what's the best thing to do in this situation, and it would point me at a algorithm.
And when it comes to fine-tuning those things, then it takes a little bit more trial and error sometimes with how many convolutional layers and different settings within the deep neural network. And there are a lot of, I typically use Python. There's a lot of neural network packages where you just call the network, and you can try different ones and see what works better, and then loop through many different parameters and see which gives the best accuracy.
It's a strictly empirical trial and error thing. We don't have any rules to guide us in this. Is that true?
I'm used to things like that. It's like cooking. You try and sometimes it works. There's no kind of a hard theory behind that tells you this is what it worked for this kind of problem. At the end, it's trial and error.
Someone told me once that it's more of an art than a science. I think that's true.
Well, good. Science is more of an art than a science anyway, of experimental sciences. So, but I think if I was trying to identify cells in a section, you wouldn't tell me to use a recurrent neural network, would you?
No, I don't think so. No.
So why not? I mean, that's where I'm trying to go. I'm sort of trying to go, how can we parse the options up so that we don't have to try and error every one of the many different named algorithms.
Yeah, I mean, if we're talking specifically about computer vision, then it narrowed what we're looking for a bit more. Versus if I'm doing more neural networks or designing an algorithm for like analyzing electronic health records or something, or a time series, then you might look more into long short-term neural networks or something like that.
So I'm curious about, you showed today that the spiking neural network, we usually think a spiking neural network are better in energy consumption, but it shows that sometimes they are better even in accuracy. So that's new for me.
I don't know if there's anything specific about them. I mean, it would just that scenario in comparing to what the state of the art was. We were able to get it to be more accurate. I guess who's to say if we had tried to build a recurring neural network for that case, could we have gotten that more accurate? I don't know, but that wasn't the goal, was to create something that could consume less power.
How about, could one of you just say what a spiking neural network is as opposed to a not spiking neural network?
Okay. Just if you want to talk. So spiking neural network is where the input is not a continuous input of data. You're waiting until the data reaches some threshold to then run the algorithm, and so you're running it less frequently, less continuously, so it's using less energy.
So it's like if I have an image, I'm trying to identify something, it's not moving at all. It's just an image. So that time is not a factor.
Yeah,
right? So I guess I wouldn't use it for that because time isn't a factor. Or do you make time a factor somehow artificially?
I've only used it with the time-based applications, but I mean,
that makes sense.
But we also had talked about a paper on spiking neural networks for point clouds. So yeah, if you make it a factor, then maybe you can still use them.
You'd have to, I don't know how you make it a factor, but let's say I have a time varying input and I have a time varying output. And so I'm looking for to train a machine that will give the right output for all the different inputs that it might receive, right? So I can easily think of an application for something like that. Like, imagine I'm just going to translate speech. And speech is going along however it is, and I know what the translation ought to sound like, and I just try to make a neural network that will do the translation without understanding the languages at all. Maybe that wouldn't work, but it's a cool idea.
Yeah.
And it's a time-varying input, time-varying output thing. So then I turn the input into like discrete.
It could be like
samples.
Words in that case.
I see. Okay, great. And those are discrete, and they're happening in time with spaces in between them. And then normally in a neural network, I'm imagining a bunch of different channels of information converging on neurons. And so what are the channels in an example like that where there's time? Because I'm thinking of, if I sample a time series, at any one time, there's just one value.
Yeah, so you have to quantify all of your inputs somehow. So how do you quantify sound or speech, even within the one word that you're sending it? It's not something I'm experienced with, but I do know, sound carries at different frequencies. So maybe the frequency level of your voice going up and down, if it can tell what word it is from that.
They sort of envelope and then spectral content and all of those become features of that word. that can be separated out and become different inputs. Is that how that would work?
Right. And then you'd need the translator as well. So you kind of need the identifier part of the algorithm to say what word did the person say, and then the translator to put that into the output voice.
So I'm thinking about that. I'm thinking about this specifically because neurons, if you want to, we sometimes give an input to a neuron and then we look to see when the neuron fires. And you could say, well, I mean, the perfect description of a neuron is I find the thing, the equation or whatever, set of equations, that when it receives this input, it gives this output, and when it gets this input, it gets that output. And now you have a perfect neuron spiking predictor, which is like what neuron modelers would really like to do. I'd like to have an accurate representation of a bunch of neurons. I can wire them together and see what they would do. Because normally neuron modelers make generic neuron representations that aren't actually accurate representations of any neuron. They're sort of the things that all neurons have in common. And then they make networks of those. I'm not talking about artificial neural network. I'm thinking about real brain network that we're trying to understand how it works. But if you could make your brain network that you're trying to understand how it works, make your computer simulation out of neurons that have exactly the same response to input as real neurons do, then you could make a better model of your circuit. So we could give some input waveform to a neuron and look at when it spikes, and then give another one and look at when it spikes, right? And we could just collect enormous amounts of that kind of data and try to train a machine learning algorithm to predict spike times. So that would be a good use for this, I guess.
Should I be taking notes? That sounds like a
good
project.
If you get interested in the problem, I think it's an interesting problem. But I'm just thinking, there's not really words in the input. It's just an arbitrary time series. And so one possibility is you think, well, at any one moment, I could look what that is and then give that as an input to a neuron, but that's just a one-to-one relationship, between, it's not like you have many inputs coming to that neuron. It's just this one time series.
But in real life, don't you have many inputs going to neurons?
It's true, but in the experiment, you're like saying, imagine this waveform is the collective outcome of all those many synapses. Because at some point, the current from all those synapses has to make it to the spike trigger zone. And it's only the current that does that controls spiking. And of course, there was a super complicated process that led to that you don't understand very well. But if you want to understand spike production, and not necessarily all that dendritic processing, then you could just say, okay, whenever these currents happen, then the spikes happen. So I know Erick has worked on this problem. I worked on it.
Trying to predict the response of the neuron, kind of the transfer function of the neuron. Whenever it receives this input, what is the output? And we had tried a neural network to predict it, and it doesn't work that bad. It was good, but it's not explanatory of what the neuron is doing. It's just reproducing what the neuron is doing.
Which is good enough if you just want to use it in a network simulation. In fact, it would be a great leap forward if it was really accurate. Yeah, so anyway, there are problems like that just come up that I can't immediately see how to structure a network to do them. Like what we did for what you were just talking about was we broke up the spike train into a sequence of intervals and then just matched the input during that interval with the duration of that interval. And there it really is categorizing that input. This is the kind of input that give you a 20 millisecond interspike interval. This is the kind of input that give you a 50 millisecond.
Yeah, we were given the input to the model and trying to predict the timing of the spike. We have the timing of the spike of the neuron, and the network will try to fire at the same time. And that was the loss function, the error, how far away from the real spike the network predict the spike.
But to do that, and of course you could test it by taking a bunch of other interspike intervals and waveforms and seeing how well the prediction works. But you really want to just be able to feed the thing the arbitrary waveform, not have chopped it off at the edges of the spike, and let it pick where all the spikes are going to come. And so that method didn't work because the inputs to, I guess I didn't necessarily want to talk about our problem, but now I do. So the way we structured it was you say, well, we'll break up the interspike interval into 50 sub points and times, and we'll total the current that arrived in each of those 50 points, which isn't necessarily a lot. So you have to resample the input pattern and also into 50 sub epochs. And then each of those input patterns for each of those 50 epochs will become an input to a neural network. And then the output of the neural network will be the time of the spike. And so that worked great, actually, at predicting the time of the spike, amazingly well.
Yeah.
The problem with it is that if you wanted to now use that neural network to predict spike time in a free-running way, you have to know the interspike interval that you're predicting so that you can chop up the input into the 50 correct epochs, right?
Did you ever run into issues where splitting into those 50 epochs, you ended an epoch like in the middle of an input spike?
Yes.
Would that be a problem?
Yeah.
Okay.
Yeah, so it's there must be a million issues like this where like the biologist with a bunch of data goes, wow, I don't really know how to get started here because I downloaded some neural network software and it's basically a classifier.
Yeah.
So it says enter your measurements here. You can say how many of them there are going to be. There has to be that many of them for every trial. And because these are the attributes of the thing you're classifying. And then you have to tell it, here are the possible outcomes. It could be this, or it could be that, could be that. There may be a lot of them. In the case of interspike intervals, it was continuously distributed, so there could be really a lot of them. But most of the time in a classifier, it's like, what cell type is this? How many cell types are there? Well, we'll go with no more than 20 cell types, say, or something like that. We'll find a 20. And so, and then to train it, you have to say, this went with this, and then this goes with this, and this goes with this. And so breaking your problem up into that landscape is the first barrier, unless you have a really whatever canonical problem for classifiers. Yeah, anyway. Maybe I've taken us into territory we don't know any of the answers to, but...
I mean, it's a lot of data labeling at first, for sure. And takes experts in that field. So that's less neural network development and more like what's the tedious task that you're trying to automate.
Yeah, prepare the data to start running the neural network is maybe half of that work or something like that.
So when you get that, you get it prepared for you. Like you've got some biologist says, this is my data set. I want you to work with this. then you look at that data set, and I know you said trial and error, but I don't think you're just going to like sort of randomly start trying things in no specific order. Probably you'll look at it and you have a hunch about the right way to proceed.
A history of like using segmentation versus key point detection, classification algorithms. So we have these like subcategories, and everything within that is more similar. So the subcategories of neural networks, and then you have to also take into account what the goal is of the neural network. Is it just to identify all of the objects in this picture, or do we want to classify them? Do we need to know their area, or is it good enough just to put a box around it? So all those are things that we take into consideration as well. And that affects how you label the data, the input data too.
So the categories of neural networks that I know about, which isn't all of them for sure, but you don't mention them. Like you don't say, well, I would always use a statistical kind of method for this. I would always use a neural network for that.
No, I mean, I tend to choose something maybe that's simpler to start out with.
What's the simplest?
I mean, for computer vision, sometimes you don't even need a neural network. If you go into like the watershed method or thresholding, then it's kind of forcing everything to either be black or white. So it's taking out all the grayscale from the image. And I've worked on projects where that works great, and there was no need to make any complicated convolutional layers or anything.
It's sort of 1980s style image analysis.
Yeah, and then you can, add in, we call it rule-based algorithms as well. And so that's really just, you're telling the algorithm every time this happens, I want this to happen. So say I'm trying to detect cells with the watershed algorithm. It pulls up a bunch of the cells in white, the background's black. Great, those white circles are cells. And then you have a few small, like white artifacts in the background that are clearly not cells. You could create a rule that says if the identified cell is smaller than whatever threshold, that doesn't count. And then you're left.
So it's got to be at least K pixels or throw it away.
Right. Or a diameter, or some other way of like measuring the shape or something like that. That would be.
Yeah, those sound really simple. Those are the kinds of things I was trained in doing just as a microscopist with a computer decades ago.
Yeah, but even then, I mean, the algorithm itself is very simple to implement, but if you tweak different threshold patterns of what you want to count as the cell and what you don't want to count as the cell, then that can completely change everything. And then if you want to get into classification, not just identification, then you still need that more in-depth information. So it would hurt you to change all the pixels to black or white in that case.
Yeah, definitely. I mean, I've got to say, I don't think those really simple methods were very satisfactory. That's why we're so excited about
more
machine learning algorithms.
Yeah. So like the next step up example would be like the deep neural network UNet that I referenced earlier today. It's just, it's fairly simple to implement, encoder and decoder, and that ended up working very well for our purposes.
50 of what?
50?
Did you not say 50?
I don't think so.
So I didn't understand, I guess, the name of the neural network that you were talking about.
UNet? I'm actually not sure where the the name comes from, but it was designed for identifying.
And that's a
cellular level.
convolutional neural network? And not spiking neural network?
No, not a spiking.
Convolutional neural network.
Yeah, that's a very interesting architecture of neural network that was created specifically for images. And you have an input layer with small network that's gonna read a part of the image, and every little network in the input layer will convolve the image and put the data in the output layers, and it's gonna go as always in the neural network to the output. But the first layers are convolutional, are convoluting a little piece of your image, and
it's like with generalizing.
This is, in neuroscience we call that a receptive field.
Yeah.
And each little place is got a little receptive field, and so it's responding not just to a point, but to a pattern in its little.
Yeah, and there is the analogy that is kind of the first layer is kind of like the retina. Then you have V1, V2, V3 until the classification maybe in the frontal lobe or something. But it's much more simple than the brain, because you can see how it's built. Every connection is known. The full architecture of the network is known. It works kind of like the visual system.
So I get the argument. The argument is the visual system uses these kind of receptive fields to find contrast on a certain spatial scale. And so I'm going to add that into my neural network because the brain does it or the visual system does it? Or is there a better reason than that? What do you get out of that convolutional process?
You get if you're the center of the convolution is the pixel you're focusing on, you're getting information from the surrounding pixels every time you complete the convolution.
So do you want all the surrounding pixels, or you want the whole? I mean, why not just use the whole image then? Because then you get information from everywhere.
Yeah, but something further away from one object on an image might not be as relevant to it as the neighbors of that pixel. That's where the border information comes in.
I guess I'm just sort of asking practical question, how big should be your convolutional kernel?
This is what that's the
is
trial
error thing. Just tweak
the size, how the stride length, so how far you move the kernel over. That's all things you kind of play with. I mean, there's some standard starting points, I guess, but in a range, you typically wouldn't make the kernel almost the size of the image or anything like that. But yeah, a lot of it is stuff you just play around with.
Okay, well, I'm ready to just start trial and erroring. I thought it was going to be a lot more complicated than that. So thank you very much, Courtney.
Yeah.
And Eric.
Thank you, Shelley.
This has been Neuroscientists Talk Shop.